Hello, my dear students. Today I will be talking about Biology Cambridge IGCSE Paper 2, multiple choice questions, and it will be extended part actually. The subject code is 0610, as you know, and the component code is 22. I have chosen the latest paper, May-June 2026, so let's see how can we go about it. As you know, each question carries 1 mark, and we'll try to see the main strategy of exclusion of the option so that we can jump to the correct option. So question number 1 says, a man moves from a sitting position and walks towards a closed door. He opens the door and takes a short walk. So if you see, he was sitting somewhere, and then from this to this, there is a change of position according to the surroundings. Looks like we are talking about sensitivity. He opens the door and takes a short walk. Which characteristics of living organisms are demonstrated by the man? So if you look at the options A, B, C, and D, of course, he's not growing anything, is he? So it's not the growth. Let me take you to your book just for a while. Look at this. Growth. All organisms begin small and get larger by the growth of their cells and by adding new cells to their body. So are we growing new cells to the body? No. Growth, so B and C already we will try to exclude. We will exclude it rather. What about nutrition? Is there any scenario where the question is talking about the man is eating something or he has some food, etc. We don't see any such thing. So we are excluding this also. Now, our correct option is A. But why it's correct? Excretion, respiration, and sensitivity. How can we justify that? Justify that? Excretion is the removal of the waste after the metabolic reaction. So what, for example, in this situation, what is being removed is carbon dioxide gas, which is definitely he's respiring, and when for respiration he needs energy to get up and go and open the door and go outside. So excretion, respiration, and sensitivity are serving the purpose. Why sensitivity? Yeah, this could be a little, what should I say? Um, Question mark for the students. Like, he's not responding. No, he is responding. He was sitting, and then he is going to change his position and opening the door and going for a short walk. It means in his first his surrounding was sitting somewhere. And then when he started walking, the surrounding is changed. So sensitivity. So A will be the correct option. I hope it's okay. Question number 2. The bar chart, the bar chart. So if you see, this graph is actually comprised of the bars, so we call it bar chart or bar graph. Shows the number of arthropods and the types of arthropod in 1 square meter area. 1 square meter area. 1 square meter area means 1 meter, 1 meter. So 1 square meter. So this is called 1 square meter area. So meter by meter. So if you go for simplification, 1 meter, 1 meter, 1 meter. So square meter. Look at that, the graph. If you look at the x-axis side, you have 4 main types of arthropods. You have arachnids, the best example, spiders, as you know. Then you have crustaceans, crabs, for example. Then you have insects like flies, mosquitoes, cockroaches, etc. Then you have myriapods like millipede or centipede, etc. Anyways, types of arthropods, and if you look at this side, which is y-axis, at y-axis you have number of arthropods. So in a way it is very easy bar graph. You can see easily the insects are the highest one. Why? Because if you take it back, it's 16. It's touching 16 somewhere over here. This part, if you see, 16. So how many arthropods? 16. Insects. So this is the highest one, as very simply we can say. And then you can have crustaceans, and then you can have arachnids, and myriapods are the least one. But most probably this is not our question. The question says, how many of these arthropods have more than 3 pairs of jointed legs? Very interesting. Now you don't have to say which has the highest number of arthropods or which are the least number of arthropods. Rather, you have to talk about how many arthropods altogether have more than 3 pairs of jointed legs. So if you go back to your syllabus, you know we have 4 main types of arthropods, and the 3 pairs of jointed legs is the ones we have for insects. Looks like if we exclude insects, the rest we can add them. So add them. You add 1, 2, and 3. So how many arachnids we have? We have 10. How many crustaceans we have? We have around 12. Yes, it's 12. And how many myriapods we have? We have 5. So how many altogether? 10 plus 12, 22. 22 plus 5, 27. So 27 will be your answer, for example. Of course, you're not going to take this much log in your examinations or in your practice, but since we are discussing the paper, so I'm going slow for that. Number 4, which says a leaf is viewed under a microscope. The thickness of the leaf is measured as 6 millimeter. When you are viewing something through a microscope, of course, it is an image of the specimen. And the thickness of the image is 6 millimeter. The magnification used was 40 times. The magnification of the microscope is 40 times. So you have been given magnification. This is the magnification M. And this is image size. what is the actual thickness, which is A. Actual thickness is A of the leaf in micrometer. So if you look closely, we have been given the image size as millimeter. It means we need to convert millimeter into micrometer. Millimeter into micrometer. So when we convert millimeter into micrometer, we need to multiply by 1000 because 1 millimeter is equal to 1000 micrometer. So I think it's going to be simple. 6 millimeter multiplied by 1000 will be equal to 6,000 micrometer. It will be 6,000 micrometer. And now you can put the formula. And as you know, the formula is magnification is equal to image size divided by actual size. But as you know that you have been given magnification, you have been given image size, you need to know the actual size. So if we modify the formula, it should be something like A, which is the actual size of the leaf, leaf thickness, is equal to image divided by magnification. So you just need to put the values and you'll be getting answers. Already you converted millimeter into micrometer. It means when you will have the answer, it should come in micrometer. So just put the values. 6,000 divided by 40. So of course, you are getting 140, which is D, and your answer is D in question number 4. Okay, so we are now question number 5. Question number 5 says, potato cylinders were placed in sugar solutions of different concentrations. So you have some potato cylinders and different sugar solution concentrations. The lengths of the cylinders were measured before and after they were placed in the sugar solutions. So already you measured the length before and after. The ratio of the final length of the potato cylinder to initial length was calculated. The initial length of the potato cylinder was 4 centimeters. So if I try to make it simplified for you, I'll go just by making simple diagrams. You have you have 5 concentrations. So if you see 0.1, 0.2 moles per cubic decimeter, 0.3, 0.4, 0.5. So it looks, we have 1, 2, 3, 4, and 5 beakers. And you have potato cylinders 1, 2, 3, 4, 5. And all these potato cylinders, the initial length is 4 centimeters. So it's 4 centimeters. Now you have put the potato cylinders in each sugar solutions, and you need to find out what was the final length of the potato cylinders that was placed in zero point 0.1 moles per cubic decimeter. Looks like you need to know about this. You do not need to know all um, about all the solutions, but specifically this one. But before that, of course, we need to do some calculation part. So on x-axis side, you have sugar solutions. Let's study the graph and then we will go ahead. If you look at the x-axis side, this is x-axis side. X. This is x-axis side. And you have 5 sugar solutions, 0.1 cubic moles per cubic decimeter, 0.2, 0.3, 0.4, and 0.5. If you look at this side, which is y-axis, so you have 0.8, 0.9, 1, 1.1, 1.2 ratio. It's a ratio of the final length to the initial length. So in fact, there is a formula for ratio calculation. And what's that? It is this kind of calculation you should know that the ratio is equal to final length of the potato cylinders divided by initial length of the potato cylinders. As you know, initial is the same for all of them. It's the same. It's 4 centimeters. But the final length definitely is different. And you have been given the ratio, you have been given the initial length. So you can calculate the final length. Of course, you need to calculate that one. If you look at, because they are not asking you all, they are just asking you 0.1. This part they are asking you, this one. So of course, you put the values for ratio for final. So we will modify this formula. This formula we will modify. So if I bring this formula this side, so it should be final length of the potato cylinders is equal to, this is a simple math. As you know, in biology, we do not go, usually we do not go for very complicated math. So simple math, just a change of values here and there, and then you get the formula modified. So final length is equal to ratio multiplied by initial length. So what is the initial length? Already you know. What is the ratio? We need to know from the graph. If you look at the graph, the 0.1 ratio is 1.2. So 1.2 multiplied by 4. So when you multiply 1.2 by 4, you need to calculate what you are getting. Is 4.8 centimeters. So we also calculate, it will be 1.2 multiplied by 4, it will be 4.8 centimeters. And if you know, if you can see the options, you have 4.8 centimeters somewhere over here. So B is your answer. I hope it's clear. Okay, question number 6 says a DNA molecule consists of 2 strands. As you know, DNA has 2 strands. This is going to be also very simple. Which row shows 2 bases? And please keep in mind, we are talking about DNA. We are not talking about RNA. We are talking about DNA. So when we talk about DNA, actually we talk about adenine, guanine, cytosine, and thymine. AGCT. These 4 nitrogenous bases we talk about. Okay, so which row shows 2 bases that form bonds that hold the strands together? So if you look at base 1 and base 2, option A says AC. A never goes with C, never. So wrong, excluded. GA. G never goes with A. G with the T. G never goes with T, or T never goes with G. A with a T, or T with A, of course. So the bonding is every time between A and T, adenine and thymine, and cytosine and guanine. You know that. So D is your answer. I think it was very simple, I hope. Question number 7. Which statement about enzyme action is correct? Now, you need to know enzyme action, and you have to justify which statement will be correct. Above the optimum temperature, the kinetic energy of the enzyme and substrate decreases. Do you think so? No. It keeps increasing, of course, because the more temperature you give, the more kinetic energy will be there. B. The enzyme maltase has a complementary shape to starch. I don't think so. Maltase will have complementary shape with not starch, no starch. It will be maltose because it is so specific to maltose. So B also we are excluding. A excluded. B excluded. Number C, the enzyme pepsin denatures in all acidic pH values. No, as you know, pepsin works best in our stomach, and in our stomach, the pH is actually acidic. So, no. So, only option which we are left with is D, which should be correct, of course. The frequency of effective collisions between the enzyme and substrate is greatest at the optimum temperature. Of course, it's correct, because optimum temperature is the best temperature for the collision of the enzyme and substrate molecules, and it gives the best results.